My name is Joseph Strangis, and I'm going to give a presentation on small molecule activation at synthetic iron-sulfur clusters. So, iron-sulfur clusters are these extremely ancient, small inorganic protein cofactors. They're found in almost all organisms. They have an iron atom coordinated by sulfur atoms found acting as these small actors within cells within the body, and their key function is electron transfer as in redox reactions. So many iron sulfur enzymes contain the cluster near or at the active site, which would indicate key roles of these cofactors in catalysis, especially since protein structure is a big determination of their function. And these clusters are known for roles in not only redox reactions in cell organelles like the mitochondria and chloroplasts, but they're very useful in nucleic acid metabolism. So they're pretty much hallmarks of cellular respiration almost. Geometrically, they come in all different shapes. At the right, there is a rhombic shape shown in A and a cubic shape shown in B. And there are a few different types. There are 2Fe2S clusters, which have a rhombic structure with 2 iron atoms. They're typically ligated by 4 cysteine residues within the iron-sulfur protein, but the ligation involving both cysteine and histine residues are also observed as well. And they're found in a bunch of proteins that are involved in synthesis of things inside the body. Another type are 3 iron-4 sulfur clusters, which are a little less common among these types, but they exist in a variety of proteins still, primarily ferredoxins, which are comprise a lot of iron-sulfur clusters, and their specific coordination in iron-sulfur atoms is a unique arrangement. 4 iron-4 sulfur clusters have a cubic structure with iron and sulfur atoms found in alternating corner patterns. They're coordinated by these cysteine residues themselves, and they're found in some bacterial ferredoxins within the mitochondria. And these all have different roles. Iron-sulfur clusters play crucial roles in different biological systems. They're very versatile because of their electronic properties. They help out in things like photosynthesis, respiration, DNA replication and repair, and gene regulation. And they help doing these things in terms of electronic transfer, where these clusters are components in electron transport chains, and they participate in redox active centers for proteins. Enzyme activity, where the clusters are involved in the regulation of enzyme activity. They are crucial for binding and activation of substrates and enzymes, and they're involved in the transcriptional and translational gene expression of bacteria, and they're strong structural units in proteins like ferredoxins, and they're also serving as storage units in those as well, in organisms and for the proteins. So they're very useful complexes. They're very unique in how they function. So it is a very popular research topic to synthesize clusters around a specific design. So these structures are engineered for specific reactivity towards smaller molecules, which enables an activation and conversion of these molecules into different products. So synthetic iron clusters can catalyze reduction reactions in all different forms. In this diagram, we see different schematics for these clusters from left to right. We see 3 iron, 1, 2, 3 with 2 sulfur arrangements. Here we see 3 irons with only 1 sulfur arrangement. In these two here, we have a combination of 4 iron, 4 sulfur clusters. We even have iron 4, sulfur 4 clusters in the diagram to the right, which we'll talk about a little later. And these structures themselves are emerging as transcriptionally regulated mediators in disease, but they play a crucial role in just our normal physiology of the organism, the cell, taking care of itself. So they offer potential new therapeutic targets for all kinds of diseases, specifically relating to malaria, diabetes, and some forms of cancer. And when we design these clusters around the small molecule activation of molecules we're concerned about, they have a whole new wide variety of ways that they could function. So small molecule activation itself is the transformation from a simple structure to something larger, maybe into something like a polymer. And they're found, iron-sulfur clusters are found in the conversion of carbon dioxide, carbon monoxide, and diatomic hydrogen gas conversion. And the activation is catalyzed by these clusters, often through these redox reactions, where electrons are transferred between a cluster and the small molecule substrate itself. But they could also be designed to mimic activity of enzymes that aren't necessarily iron-sulfur cluster-based enzymes, maybe nitrogenase or hydrogenase. And they possess these specific structural and electronic properties that help facilitate this activation. So this comes to build a question in science where, now that we know that these properties exist, how do these physical properties themselves contribute to reactivity of iron-sulfur clusters? So we're going to take a look at some research done by Daniel Seuss, who specifically looks at iron 4 sulfur 4 in its empirical form clusters. And he uses these because they're very versatile cofactors that help facilitate electron transfer in all kinds of respiratory processes to maintain homeostasis. And these particles participate in nitrogen fixation, DNA repair, just as we've seen before. But they also serve very important intermediates for enzymatic reactions. And we see that when specific alkyl ligands excite the structure of Fe4S4 when bonded to its corners. Here we see some examples of these structures with groups attached to it on the right. So by investigating the electronic structural properties of these alkyl complexes, we could get insights into the behavior of these intermediates and their role in the enzymatic process. So the clusters that were synthesized in this experiment are shown in the middle in 4 and 5. We have the synthesis and characterization of a 3-to-1 site differentiated iron 4S4 chlorine and ethene cluster. In the left, the figure shows insights on the electronic structure of the synthesized Fe4S4 clusters with the ligands, with the alkyl ligands. The picture shows a simulation of the Mossbauer spectrum with the clusters, indicating the distribution of electron density among the iron sites. The simulation suggests kind of a 2-peak electronic structure for the clusters, with a ground state of 0 arising from the antiferromatic coupling of the two rhombs, each containing spinaline high spin iron atoms engaged in the double exchange interaction. But we could also see from this figure the assignment of spectral features of specific iron sites within the cluster, indicating similar core charge density and formal oxidation states in each iron site. We also see the symmetry of the ligand-derived resonance observed in the NMR spectra of these clusters. To see more of their structural properties and electronic behavior, we could divide two peaks by putting a line in the middle. On the right, we see the results of the broken density functional theory calculations conducted to analyze the electronic structure of the clusters with the alkyl ligands. The figure represents the diagram showing the localized molecular orbitals and the distribution of the electrons among the iron sites of the clusters. The localized molecular orbitals are partitioned into a set of spin up and down iron 3D orbitals, which an additional delocalized orbital mediating the double exchange interaction between two iron sites. This configuration matches up with the same electronic structure that we see on the left, where the inherent electrons are fully delocalized, leading to a conformal oxidation state assignments of each iron site. Now, we're going to explore controlling the substrate binding to iron 4s4 clusters, because the reactivity is influenced by the protein scaffold of the substrate with these working organometallic enzymes. And coordination can actually be regulated by steric effects. And one in particular are N-heterocyclic carbene NHC groups, which tend to be very big and take up room around the structure. So the reactivity exhibited by many iron-sulfur enzymes is not only due to the scaffold, but we can control remotely the activation of binding sites through steric effects on ligands. In this paper, we're going to look at constructing or synthesizing a 3-to-1 site Fe4S4 clusters with these sterically hindered ligands. And the results tell us, on the left, the figure shows the molecular structure of Fe4S4 clusters with the NHC ligands. The figures compare the structures of two clusters, and they lead to site differentiation and generation of the different substitution locations. The comparison highlights the impact of the steric effects on the coordination chemistry. So we could see how these different grade groups, where their locations are, we could see how remote steric effects influence the site differentiation within the cluster, and how it can control the coordination environment of the iron sites. The figures on the right show the EPR spectra and the UV-Vis spectra of these clusters with different ligands, showcasing their structure and spin states both. We see data on clusters with NHC ligands with different steric profiles, and we see ground state spins of positive 1/2 for the clusters, which is consistent with the previous observations. And the UV data provide additional insights into the electronic structures of the clusters, highlighting the impact of the ligand steric properties, which tends to dip a little bit. So, because of how widely used these clusters can be, we can use different properties from steric effects, electronic effects to make new iron-sulfur clusters that can either catalyze all different kinds of reactions, but could also help facilitate metabolic processes to maintain homeostasis better. These could be great drug agents, or they could be great treatments for certain diseases. We've seen from the research achieving site differentiation within the iron 4s4 clusters can control substrate binding to materials we're curious in, affecting the small molecule activation regarding these clusters, and their electronic properties can be studied to design around these molecules that we are concerned about. Here are my references for my presentation, and I want to thank you again for listening.